and the row. Just moving. So this is, you can see all the senior staff members now. They were youngsters back in the day, front seat, and you will see me in the corner. Just moving the me in the corner, and Madam Kumar and Kumar sir in the middle, the department head and the dean back in the day. Ile Perumal sir, I can see, and I met him a couple of years ago in Boston, and also HM Bandar sir. Looks like Rajpaksa sir and Champika madam is not here in this picture, but rest of the senior faculty, I believe here. Namal sir, Ayanti madam, Chandani madam, Anurag sir, Atul sir, Veranjan sir, two Bandara sirs, and so on. Okay. Okay, so from Peradeniya, I entered to the University of Minnesota in 2005 and studied the various aspects of carcinogenic DNA adducts. So I made them and I studied them, so mostly organic and analytical chemistry related. So I'm happy to share more on the research work if any of you have questions, want to know. But for the time being, I will skip those studies. And in 2010, I joined MIT as a postdoctoral associate and studied the chemical biology of post-translational modifications. So particularly in the area of S-nitrization. So the post-translational modification is the protein is changing after RNA transcription, adding a phosphate group or adding a NO group, or because of oxidation to a cysteine. So in 2015, I joined Pfizer neuroscience group. Can you guys see my screen well? Because I can see the pictures coming.
That's fine. That's okay. Yes, okay. We can see well.
Yeah. Okay. Thank you, Anurag sir. So in 2015, I joined Pfizer Neuroscience Chemical Biology Group. Now sits in the East Coast Chemical Biology Group at Pfizer Cambridge site. So we are helping advancing in number of therapeutic areas, cardio, metabolic, inflammation, and immunology, and also rare diseases. And we are commitment to innovations and breakthrough science to the well-being of our patient. So that's actually the new logo describes. Okay, so this is a picture, aerial picture on MIT. And so we are located at Kendall Square, the heart of Cambridge City, Massachusetts. And the Cambridge is dubbed as the Silicon Valley for the biotech and biopharmaceuticals. So this includes more than 430 biotech companies, including startups and all major pharma in within 2-3 miles in Cambridge City. So the picture is MIT, and a block away you may see the red color circle. So those are Pfizer building. I think at that time one of the Pfizer building was only one was there, and you may see to the right two Pfizer buildings, so which I am located at. So it's just one block away from MIT. So I spent in Boston. I was living in Boston since 2010. Okay, so what we do at Pfizer? So we do drug discovery and development. So a few brands you may see. I believe some you are familiar, some are not. And as may you have heard, we do some small molecules, pill or oral available pill. For example, Viagra, which is famous, and Lyrica, and proteins. I think I put a picture. Preventive 13. It's a protein based on diphtheria protein, diphtheria pneumococcal bacteria, and now famous is the RNA vaccine COVID-19. So how we do this drug discovery? So this figure is much simplified version of the drug discovery process, which span actually more than 10 to get an approval drug is take like more than 10 years, starting the project to the launching of a drug. So many projects, important thing is many projects fails, and so it is costly. So it's so risky job, risky science, I would say. And approximately 1 billion we are investing for a single drug to the launch. So the drug discovery process begins with the identification of a medical need, with the knowledge of biology, efficacy, safety, and how to improvements and to advance a treatment to patient. So then the research project team is formed, and give and objectives and goals are set. Then we do test selected chemical compounds in appropriate biological screens. Sometimes it's millions, sometimes it could be 1 million to 500,000 compounds we are screening. If the process is successful, we may find a hit. If it is successful, that modulate the target or the pharmacology that we are interested in. We then expand the hit to improve the potency and the selectivity to obtain a better chemical compound. So we call it we call it a more potent compound, and we call it a lead series. So we do check in vivo activity, which is in animal models, to maximize the activity with analogous compounds, analogous structures. And finally, selecting one compounds as the drug development candidate. We call it the CAN molecule, which is the candidate. Then we do safety measures with the chronic dosing. So chronic dosing means longer period of time. So this is for toxicological testing in animals. So we do mouse, we do rat, we do dog, and we do non-human primates. So if all the tests are successful, we then file a claim to the FDA. We call it investigational new drug application. We abbreviate it as IND filing for the FDA. And then if FDA approve, then we'll start the phase 1 trial, in which we reevaluate the compound in normal, healthy human volunteers. So this is for the tolerance, which we call phase 1 study, with few human subjects. In phase 2, we do measure efficacy. So efficacy means how good the drug is, and the dosing. So dosing means how much milligram the pill should be and how many times the pill should be. So like once per day, or 2 times per day, or 3 times per day, etc. And measure biomarkers for the disease. And this is done for the phase 2 is with the patients, not healthy subjects. And then we begin the phase 3. So phase 3 is thousands of patients. So then we develop a database for the efficacy and safety. How good? So this can run from 1 year to multiple years for safety reasoning. And then if all goes good, all has the good data, then we file the NDA. So NDA means a new drug application to FDA, and the FDA experts will review it. And if FDA gives the green light, then the commercial launching is done. And then the doctors can prescribe the medicine to patients. So that's the whole process. So you would imagine it is taking time, and it's number of people, number of brains behind a single drug. So could be few hundred to thousand scientists of making a drug actually. So what we do in chemicals, so how the chemical biology doing in Pfizer. So when Mark Bunage was at Pfizer, but now he's the Boston site head at Vertex Pharmaceuticals, and he found that back in the day, I believe this is a 2013 publication. So he found that 44 drug programs in Pfizer in phase 2 clinical trials failed because of the lack of efficacy. So his conclusion was Pfizer needs to put more resources on preclinical target validation. So we need to understand the target and the compounds well before the compound goes to the clinic, so we can save the money. So in other words, put more research to the chemical biology, establish chemical biology groups, and do more research on the compounds and the targets, protein targets, or the RNA targets before going into the clinics and failing. And there are four pillars uh, of target validation in chemical biology for drug discovery. So you can see four pillars. So pillar one is the compound exposure at the site of action. So it's mainly the cell permeability, how well the compound is penetrating the cell. Because we need a compound that penetrates the cell. If it is brain penetrant, the compound should penetrate the brain. And if the compound shouldn't brain penetrate, so it shouldn't go to the brain. So those are very, very important measures. So the pillar two is the target engagement, what the chemical biology group does every day. So this is the most technically challenging pillar. So we need to keep ourselves up to date with the advances in the field. Sometimes we need to innovate to get the science going. And pillar three is the expression of functional pharmacology, so which is assessing and measuring proximal biomarkers for the activity. And pillar four is the capture of most relevant phenotypic changes, which I'm not discussing. So not that important for the chemical biology. So the pillar two is the much more important pillar for the chemical biology. So so these are our day-to-day activities, typical approaches that we employ in drug discovery programs. So basically doing chemical reactions in cell. You will see what I am actually telling. So first is the target deconvolution. So target deconvolution is in other words, finding the protein target. And target engagement, where there is a potency or activity difference or disconnect between the cell assays or the biochemical assays. So we do see quite a number of times the disconnect. So we need to explain more why the disconnect and how to avoid the disconnect, which one to believe. And then we do selectivity profiling and activity-based protein profiling for protein families, kinases for oncology, cysteine proteases, phosphatases, and also serine hydrolases. And we do measure occupancy. Occupancy is the how well the compound is occupied in a protein pocket or enzyme pocket, and the number how is it well enough to proceed. And off targets, this is very important for safety, because we don't want to hit a protein target that shouldn't hit. That is because if it hits, then it becomes trouble in the clinical in the clinical stage. You will see at that time, you will see lots of side effects. We don't want all those side effects. We want to actually minimize the side effects, minimize the off targets hit by the lead compounds. And also we do compound binding site ID. So where if it is a covalent compound, we want to know which site, which amino acid actually it's labeling, whether it's a cysteine, lysine, etc. And we actually one of the leading, one of the leading industrial lab in the US who is doing all these binding studies and well resourced actually. Uh, and cysteine reactivity and ligandability measures, where we profile the reactive cysteinome, because this is most important in oncology. For most of the kinases has active site cysteine. So we want to know how active that cysteine is and how well our compounds are ligandable with that cysteine, and is our compound is not reacting with other cysteines in other proteins. So we call it cysteinome profiling. And PTM. So PTM is abbreviation for post-translational modification. So this happens after RNA is transcribed. RNA is transcribed to a protein. So cysteine oxidation, cysteine is a thiol compound get oxidation, OH, or nitric oxide, NO, or citrullination. It is a post-translational modification for arginine. So in arginine, guanidinium functional group becomes a double bond O instead of a double bond NH. And ubiquitination, which is a post-translational modification, which leads to the protein degradation. So we have a couple of programs with the protein degraders. You might hear that's one of the novel technique. Instead of inhibiting the protein, you are allowing protein to degrade. So sometimes it is important to degrade the protein compared to inhibition. So we want to understand the ubiquitination mechanisms because of that. And also in gel fluorescence and cellular imaging. So we are actually well resources for doing all these activities. And how do we do these things? Then we now come to the chemistry. So for the most of the cases, we need to catch the protein target that our compound binds in cells. So we do use click chemistry. Most of you familiar. So click chemistry is the 1,3 dipolar cycloaddition reaction between the azide and alkyne, which is copper catalyzed. And also we do copper-free click chemistry, which is inverse electron demands Diels-Alder type reaction between dienophile and diene. I still can remember Werenger's and Arnold's organic chemistry. In the inverse electron demands Diels-Alder type reaction, we are reacting with the TCO. TCO means trans-cyclooctene and a tetrazine. And the beauty of these, especially the beauty of these functional groups, are that they are so inert that they are not cross react with the cellular biomolecule. So they are truly bioorthogonal. So we can do the reactions in cell without inhibiting, without doing unnecessary measures to the cell. So we typically convert our compounds of interest to alkyne probes or the TCO probes for the click chemistry, and then we use the other side is the azide or the TCO, azide or the tetrazine. So then we put those functional group as a reporter handle. I might have two examples using that techniques. So in this talk, I would like to share 3 cases actually. Hope the time permits. If not, I will back down to 2. That the chemical biology group actually contributed, which played a major role. The JAK3 story, APOE target deconvolution, and the 3CL protease inhibitor program. So this was 5 months ago. So this is a JAK3. So JAK3 is Janus kinase 3. It's a kinase. So we developed this PF1600, so PF 0665160. So we gave a barcode for every molecule that Pfizer synthesized. We give a PF number, and when it reached the clinics, we give a generic name for the drug. So until then, we have the PF number for the compounds. So these are proved, these are disclosed structures. Otherwise, I cannot disclose. I cannot show any of the structures for any of the presentations. So you may see this is a simple molecule. It's heterocyclic compound, basic organic chemistry, and it has acrylamide. I put a blue arrow, acrylamide warhead, which selectively react with cysteine amino acids in proteins. So there are ton of cysteine containing proteins in the cell. So the question for us was how selective this compound for JAK3 compared to other proteins, other cysteine containing proteins in the cell. And I forgot to mention this compound is actually selective over other JAK isoforms, JAK2, JAK1, and TYK2. And the indication is for autoimmune disorders. So the question for us was the cellular selectivity. And because of that, and also we had the early functional indications of downstream pharmacology. So the team was really interested of this compound, try to understand if the compounds hits other protein targets besides the JAK3. So we made the propyl alkyne. You may see the structure, and I circle it in red circle. It's an alkyl group. but keeping the pharmacophore intact. So which the compound goes and interact with the protein and react with the cysteine with the acrylamide warhead. Then we can do click chemistry from the other with the alkyne function and try to pull out the protein from the cell and do the typical workflow for mass spectrometry, etc., to identify with these what these compounds are binding. And in here, we did use to evaluate the target occupancy, how well this compound is occupying in a target or targets of targets. We use human PBMCs. We did, I believe, 5 human donors got the PBMCs, like cells, number of cells in the bloodstream, and put those compounds and the clickable probes and enrich. Did the click chemistry and enriched for the protein targets that these compounds are binding. And then we did the TMT 10-plex mass spec based assay. In here, we actually can do multiple doses in one single run. So you can see a nice dose response curve to calculate those E50s. Okay, and I was the one of the leading people who bring this technology to the Pfizer. Okay, so we found that not, you may see, we found that the bottom message is that the compound is, it's actually selective of other JAK3 JAK isoforms, but it is hitting multiple other kinases. So it's, then when we carefully looked at these kinases, actually they are belong to a single family. We call it TEC family kinases. We did see 5 of the entire TEC family kinases hit by these compound in 100 to 400 nanomolar OC50 range, which was actually interesting. And also we came to know that all these protein targets have an analogous cysteine site, so the compound can react. Besides that, there are no other kinases or other off targets these compounds are hitting. So after this surprise finding, we next went ahead with an in vivo target occupancy, because we want to understand it is not only in cells, it is actually in mouse models, because when you administrate these compounds, a lot of things can happen. For example, metabolite formation, a lot of things can happen inside the animal body. So we did a single oral dose of, so it's a pill. So we gave the pill to the, we mixed the pill compound with the food. So we give 30 milligrams per kilograms of mice. So we measure the body weight of the animal. So then we know how much compound we can dose it to the human. So these are very important indication. Here we did 30 milligrams to the NSG mice. So these are humanized immunocompromised mice. And we engrafted human PBMC cells to these human PBMCs to this mice model for 3 weeks. And then we measured, then we killed these mice and got the spleen. So spleen has all these PBMCs and mouse PBMCs as well as human PBMCs, and did all like time-wise 0.5 hour to 2-day experiment to see how much the target JAK3 and other targets is occupied these compounds. And actually we observed 100% target occupancy. We were surprised for all the early time points up to 3 hours for all these TEC family and JAK3 kinases. And later the compound was cleared off because of the metabolism, animal metabolism, and also the protein resynthesis rates. So this data actually publicly available. So we published this in a couple of years ago in ACS ChemBio. And the message is we were able to demonstrate the in vitro system to in vivo target profile for our compounds and build confidence and better understanding of our components, which was immense value. Okay, so the big question is how well this compound is doing in clinics, and a picture is worth than 1,000 words. You see the difference. So as I mentioned, the Pfizer because of the chemical biology data. Pfizer rebranded this compound, this drug as a mixed JAK3 TEC kinase inhibitor, and the compound is in phase 2B, actually now phase 3 trials. And we are treating it for indication is alopecia areata, so you lose your hair and also the hair in your eyebrows. But after the treatment, you may see in this picture, after 24 weeks, you can see eyebrows is nice and also a beautiful hair for the subjects. And due to this, and this is the first compound I contributed in the clinic. So it's so beautiful to see the work you have done, ultimately reaching to the patients and doing well in the clinic. So due to this unique polypharmacology. So polypharmacology is it's hitting multiple targets in the human body or the cell. So we call it a polypharmacological feature. So based on that, we actually Pfizer began more phase 2 trials for the ulcerative colitis, Crohn's diseases, and vitiligo, which are another 3 autoimmune diseases in the world. And actually, they are doing well for those 3 indications as well. So we believe that the dual activity of a PF1600 towards the JAK3 and other TEC family kinases is unique among the clinical compounds with our competitors, and provide beneficial inhibitory profile for inflammatory and autoimmune diseases. Okay, so next case study is the APOE program. So I will just briefly describe what we did here. So APOE program is based on a phenotypic screening. Does anyone have questions, or you want to ask questions after the presentation, or you can disturb me anytime and ask questions if you have any. So this program is based on a phenotypic screening. So phenotypic screening is a strategy to identify molecules with the ability to alter cells phenotype. For example, in here, so we were looking for the APOE protein secretion by adding the compounds, but APOE is not the target that we are looking for. We want another target which is leading. We want to hit another protein or target which actually leads to after a signal cascade, affecting the APOE protein secretion in cells. And why we do care about the phenotypic screening? So it is actually easy to translate these preclinical findings to patients and disease relevant patients and cell types, because we are doing it in cells rather than getting a pure protein and adding compounds, which is mostly sometimes it works, but not all the time it works. And if you and mostly if you want to find first-in-class drugs, first-in-class targets, you may need to do phenotypic screening to find those first-in-class targets, class drugs. And also high rate of failure of target-based projects due to this lack of efficacy indicates the underlying hypothesis is often not correct. So those things were eliminated, can be eliminated actually by doing phenotypic screening in right cells, actually, right system. So why the APOE? So APOE was a program in neuroscience back in the day for Alzheimer's disease, and you may heard some other industrial organization has some other hypothesis, for example amyloid hypothesis, but this is another hypothesis that APOE is strongly genetically associated with Alzheimer's disease. So how we can modulate the APOE and affect the Alzheimer's disease patient population. So APOE shows actually a strong genetic association with Alzheimer's disease and plays an important role in clearance of toxic amyloid beta fragments from the brain. And the project goal is to find compounds that enhance these properly lipidated ApoE proteins in the brain, but not in the periphery. So periphery means you don't want the protein ApoE increase in the liver, because then you will see fatty liver, which another disease actually uh, uh, um, can lead to cirrhosis. So we want to hit the ApoE in the brain, but not in the other systems. So we need to be very careful. So in our screening funnel, you may see. Even though this is one picture, it's like actually like 3 to 4 years of 3 to 4 years of work, trying to get the cell line, trying to get the assays right, try to get the multiple ends and screening. In here, we did screen around zero 0.5 million compounds. So you see how much resources in drug industry putting to get these assays work and get the right data for the drug discovery. So in the screening funnel, we did remove compounds. We started with 0.5 million compounds, and we did remove compounds that has the cell toxicity. And we did lots of counter screens to eliminate unwanted mechanism, because we want only the ApoE secretion, but not other secretory mechanisms. And also we were very careful about avoiding the increase of hepatic lipogenic genes. So we were measuring these ABCA1, SERB1, and FASN, for etc. For fatty liver indicative genes. We don't want to hit these proteins, which will become a safety problems in the clinic. So we did use these counter screens to eliminate those compounds hitting these protein targets. And then we narrowed down to start with 0.5 million, and we narrowed down to 5 chemical series. So 5 chemical series means you have very different, unique chemical functionality. We call it 5 chemical series. So we did find 5 chemical series, and I cannot disclose I cannot disclose most of the structures, but I can disclose only 2 structures for this program. And I'm grateful for Pfizer to allowing me to disclose those 2 structures. We were able to publish this study. So we did all these without actually the take home message is we did all these studies without knowing what actually these compounds are hitting, what protein these compounds are hitting. So the $1 million question is what is actually the target that these compounds are hitting. So now the question comes to us, to chemical biology group, how actually what is the strategy, what is the how to find the target these compounds are hitting. And you may see compared to the JAK3 example that I have shown. So JAK3 is a covalent irreversible inhibitor. So it react with the cysteine and make a covalent bond. But this is not a covalent molecule. So this is a non-covalent molecule. So how do we pull these compounds? How do we know these compounds interacting with proteins, and how we can see from the available technologies? So then again, we decided to save the day. So we did. You may see from the to the right side, you may see another structure, which it in red color the functionality. So we call it a benzophenone. So benzophenone is actually UV active. So we put photoactivable functional groups for these molecules, and we made hundreds of molecules because we don't know where to put the benzophenone, because we don't have the SAR, structure activity relationship. So, but we have some kind of idea where to put. But we did make all these around 100 molecules of benzophenone and other photoactivable moieties in the molecule. And you may see this also the right-hand structure, compound structure has alkyne phosphopyryl group, so we can do the click chemistry. And the parent molecule, the molecule in the right side, is around 50 nanomolar in the active assays, and the compound in the right side, the benzophenone containing compound, alkyne compound, we call it clickable compound, is around 1 micromolar. So we are happy actually with even with 1 micromolar, because you lose the potency when you add big bulky groups. With 1 micromolar, we were able to show where actually what target is actually hitting. So, and we kept you may see the red you may see in both the molecule, we keep the left side of the molecules keep intact. We call it a pharmacophore. So we keep the pharmacophore. It's the main constituent of the molecule that interact with the protein, protein or proteins. So we keep it constant, but keep the rest of the part. We keep changing the rest of the part by adding and removing the photoactivable functions. And we add both the compounds to the cell, typical chemical biology, chemical proteomics workflows, and induce shine the light, UV light. So what UV does is it activates the carbonyl function of that benzophenone and make a carbonyl diradical, and cross-link with covalently cross-link with neighboring amino acid. And we do, and it's purely, it can be any amino acid. It has a prevalence, benzophenone has some prevalence towards amino acids, to specific amino acids, but it can react with, theoretically, it can cross-link with, because it's a biradical, it can cross-link with any amino acid in the protein. So anyways, we did all these chemical proteomic workflows and put it to the mass spec, quantitative mass spectrometry. So we identified what is the protein target, and you can see from the to the right side picture with the red circle, LXR beta. So which is oxytol receptor in the it's a transcription factor. And we demonstrate overexpressing the cells. Typical chemical biology experiments, we overexpress because we now know the target LXR beta, but we want to validate that it is the real target. So we did bunch of experiments with overexpression, the protein target, and do in-gel fluorescence and endogenous target engagement with typical Western blotting. And also the SETSA. SETSA is a cellular thermal shift assay, which you can tell with the parent compound. We don't need the photoprobes for that. With the parent compound, how stabilizing this compound with the target, because you now know the target, so you can use the Western blotting to do all these nice cellular thermal shift assays in cells. So the impact was finding the cellular targets, help the project team to decide that if the project is a go or a no-go, which is also actually a good decision, because we don't want to waste the resources to a project that is not going to succeed in the clinic. So instead, we will stop the program and we put resources to the other program. So that's the industry actually works. So, and team didn't want to pursue with this target, because the analogous protein is the LXR alpha, and LXR alpha and LXR beta both are very close in structure. And if these compounds hit LXR alpha, they will. So it's like a fatty liver disease. LXR alpha is mainly expressed in the liver. So if the compound is hitting both the LXR beta and LXR alpha, it can lead into fatty liver. So because of those reasons and some other reasons too, the team decided to abandon this program, so we can put these resources to other programs. So that is the decision actually industry is making. Stop the program if we know it is a no-go, and put those resources to the other programs. So when the team decided it's a no-go, we cannot do anything. But we were lucky in here, because we were able to publish because I was leading all the chemical biology experiments here. So we were able to publish this protocol, publish uh, this study in two papers, one in the protocol paper, one in actually early this year, cell chemical biology. And also we were able to find the because crystal structure. If you know the target, you can crystallize with the compound. But we were develop mass spec-based assays to identify actually which amino acid, which protein side, which amino acid actually these proteins are, these compounds are cross-linking. And then we do molecular dynamics modeling to show that actually indeed it's in the site in the enzyme, sorry, in the protein pocket that this compound should be there. And later we obtain crystal structures, and exactly it's recapitulating the mass spec data that we obtain prior to the getting X-ray crystal structures. And so the bottom line is we actually this decision, the hitting our compounds hitting LXR beta, the kill the program actually. Okay, so I'm couple of minutes behind, but I want to give this because all maybe most of the people want to hear COVID-19, and I'm actually not authorized to talk about COVID-19, the vaccine program, and so this is the program that we were involved, so which maybe you haven't heard. So COVID-19, we call it the PF07321332. This compound is in phase 1. And this is the SARS-CoV-2 COVID-19 inhibitor. So this is a pill. This is a compound. You can take it. You can take it from your mouth when you have the COVID-19. So this is the first orally administered and it's administered compound in the clinic that targets the main protease. We call it 3CL protease of SARS-CoV-2. The virus you already know which causes the COVID-19. And you can see from the simple chemistry, organic chemistry, this has a nitrile function, and actually this nitrile is actually reacting with the cysteine. This warhead actually reacting covalent reversibly with a cysteine, I believe it's cysteine 145 in SARS in 3CL protease in SARS-CoV-2 virus. So the rationale behind the pill. So why we need a pill? So the reason is that when you the COVID-19 vaccine is a preventable vaccine, so it's a preventing the disease. So you need to get the vaccine before you get the COVID-19. But what happens if you have the COVID? So how you can minimize still the hospitalization? So this is it's like a pill. You take it, and it will help you to recover faster by getting just pill at home once you have the COVID-19. So that's the rationale behind inventing a pill for COVID-19. And what is this 3CL protein does? So 3CL protein actually it's a viral protein, and it's the first actually second step of the protein machinery that cleaving long chain proteins into parts, and so the viral can replicate by itself. So how the chemical biology help here? So we build the confidence of the target. So what we did here was we obtained the lung cells, and it's the host cells, human lung cells, and then we add the virus. So we in the lab we infected the host cell with SARS analogous SARS virus, and then we add the typical compounds, and also we had the clickable compounds of these at those compounds, and show that from the mass spectrometer you can show actually this viral, you can pull down the viral protein. So the team was really so that we can actually build the confidence in actually indeed these compounds are hitting in cells the viral target, which is supposed to be not the host targets, which we don't want to do. Okay, so this is my one of the last slides. So apart from developing medicines to needed patients, what actually we do? So we advance the science, the chemical biology group in at Pfizer. From actually we have three arms by driving external collaborations, and I do lead one of the external collaborations. You need to write a grant and get the money from Pfizer, and then you talk to a collaborator and funding them to do the research that we Pfizer thinks is beneficial for the to Pfizer to discover, so we can apply internally to drug discovery programs. And also the second arm is disclosing our findings to the scientific community by means of publications in high impact journals. So not only academia, so we can do publications, but not a lot, at least one publication per year. So we can do because we are well resourced to do that. Uh, and I put three cover arts from our research, from my research actually, which came into. I didn't design the cover arts, but my team did. But anyways, the work that I contributed was happy to see in cover arts in these leading journals, and put a couple of publications which I contributed as one of the leaders in chemical biology. And I do mentoring postdocs. I had a postdoc, and he's now in a good position. He's a senior scientist in the industry, and currently I am looking for another postdoc so we can publish more papers actually. And yeah, and that's it. Thanks for listening, and I hope you may learn at least something from this talk, and while spending your valuable 1 hour with me. And before you go, I want to show you a picture of my family. And this is my wife, Takshila. Takshila also graduated from University of Peradeniya from the chemistry special batch. And so we have two kids. One is 2 years old and one is 13 years old. And two hikes, one to Mount Washington in 2018, and then COVID hit. We couldn't do anything, and right after the COVID and after the vaccinations, we went to Maine actually 2 weeks ago, which was really good. Yeah, and that's it. And I'm ready to take any questions that any of you have.
Okay, thank you, Utpala. Thank you for the nice presentation. Any questions?
Dr. Utpala. So actually, I'm just really happy to just listen to your talk, and also I'm Ganehenage from the Department of Chemistry. So on behalf of the chemistry department, actually, I should extend my sincere thanks to you, especially for taking time out of your busy schedule just to give us a very inspirational presentation. And it was really interesting to learn how chemical biology and chemical proteomic technologies could be advanced for the drug development. I just have a little question, a very small question related to the drug that you showed there, that PF07321332, that SARS-CoV-2 drug that you mentioned. You mentioned there that was kind of specific, only that hits the virus part only, not
Yes.
the host there. So how that is that kind of specific? So what is the just functionality of this compound is just important for having that kind of special attack?
Yeah. So that is actually coming from the pharmacophore. So you can see it has a trimethylfluorine and also lots of bicyclic functional groups, and also the other end is it's a heterocyclic amide, cyclic amide. So all these rest of the molecules is the one actually leading the interacting with the protein. Then once it interacts, it actually interact with the enzyme pocket 3CL protease, it's reacting with the neighboring cysteine. So these compounds are very specific, very specific for the viral protease. Yeah, we do lots of human cells, lots of other off targets. We know that there are other off targets for some of the other programs, some of the other compounds. So that's how we do. We start with a compound, and we know at that time we know that it is hitting other host proteins. So then we optimize its a functional function by introducing functional groups and other moieties to be specific for the viral protease, not to the host proteins. So it's a step-by-step process, and we made, I believe, like thousands of molecules. Not only this is a leading candidate, one of the best molecules. But we have a couple of other series and non-covalent molecules with covalent molecules with other warheads. So we have all sort of molecules. We made all sort of molecules, compounds, but this is the only molecule that survived up to phase 1.
Okay, thank you very much. I think that our students may also have many questions, and
We have about 5 minutes left.
Hi, Utpala, this is Jailal. Thank you very much for your nice presentation. Actually, I have one general question. Now, after the COVID, before that, you told me, that to produce a particular drug, it takes, several years sometimes. But, after the COVID, within 1 year or 2 years, some drugs came to the market. So, is there any specific process behind that to come such drugs within very short period of time to the market?
Yeah, that's a really good question. So from the information that I can disclose, so especially for these compounds, for example, for this Pfizer compound 1332 compound, we have some other programs at Pfizer which we abandoned at that time. So this actually one of the hit compound for this SARS-CoV-2 inhibitor program came from 2003 for another internal project. So we have all those molecules, but we didn't disclose. So then once the right target or right target or the indication find, then we if the program is if some programs are at least somewhat successful, we can use those compounds to by eliminating a couple of years and move faster. So that's what we did here. And I know other companies, some other programs, some other compounds for SARS-CoV-2, they were in for other indications for longer time. So they know it's like a repurposing of the drug for COVID-19, except the vaccine. So vaccines, from the available information that I can disclose, so the technology, the RNA technology was there for longer time, but it was not in the clinic. So this is the first time that the RNA technology, RNA-based drugs in the clinic. So the science was there, but not in clinic. So it's then you are putting more resources and finding the good candidate molecule or candidate RNA compound to put it in the clinic.
Okay, thank you.
Did I answer the question?
Yeah, I think I got the answer. Yeah.
I think almost all the pharmaceutical companies did the same thing, but many of them failed, I think, right?
Yes.
Utpala, very happy to hear your voice after a long time. It's very interesting talk. I have a very simple question. Yes. I have a very simple question.
Yes, madam.
Now, without doing this in vivo, these rat models or that type of in vivo test, is there any other way to find this target occupancy without doing this in vivo rat models or dog models or whatever in the lab?
Yeah, so there are what you want to know is there any other in vivo models you can do?
In vivo, not in vivo. Is there any other way to determine this target occupancy?
Yeah, so mainly what we are doing is the cell-based assays. Yeah, so cell-based assays, and there are some other new ways like a lab on a chip you may have heard. So it's like a couple of cell lines, like I think like 3 or 4 cell lines grown in a chip. So they call it a lab on a chip. So they have a couple of cell lines from liver, couple of cell lines from brain, couple of cell lines from gut. So it's like a lab on a chip, sorry, human on a chip. But I haven't seen target occupancy. They were like mostly for the metabolomic studies, how well those compounds doing in the body. So they represented the lab on a, sorry, human on a chip system, but I haven't seen target occupancy is done in those systems. So it's either in vivo or either in cells. So we would like to see in vivo is hard. Even for us, it's in vivo is hard. But cell-based is the most prevalent. We and other industries doing the cell-based assays for target occupancy.
So you mentioned about patient-derived cell lines. What are they?
So you want to know from this for these programs or for general?
No, in general.
So it's patients mainly for the oncology, because for certain type of tumors you may need you cannot make the bottom line is you cannot make a cancer from a healthy cell. So it's better to get the cell line from a patient. So that's what patient derived cells means.
Okay.
For most of the cases, I would say.
Okay. Right. Thank you very much, Utpala.
Utpala, there's a question in the chat box from Professor Kumar.
Okay, how can I open? Okay. Oh, Vijay's question, if ESPF is being synthesized in large scale. Yes, it is synthesized in large scale. Yeah, we are doing phase 1. To do actually the phase 1, you need to have multiple kilograms, and we need to show the purity of those compounds to FDA, how pure they are, and well below the we should be in the standards to get the approval for phase 1 trial. So yes, we did made kilograms of those compounds.
Oh, he's asking that.
He says his mic is not working.
Not working.
Okay.
Yeah.
He's asking whether you can read the question.
Yeah.
From enzyme, I don't my isn't that difficult task is from the is it from enzyme? Okay, so the question is for a protein, if the is it for a vaccine? Yes, you need to make large scale. But for PF1332 is a small molecule, so you can make it kilogram. But if it is a vaccine, yes, we need to make it in multiple large scale, and Pfizer has the ability to make all these proteins as a vaccine, or for example mRNA vaccine for COVID-19. We have the capability to make multiple in big amounts for phase 1, 2, 3 studies. Did I answer the question, sir?
Yeah, Professor Kumar's mic is not working.
Yeah, he can't answer. And here's another question.
Yeah.
In order to
that is Sanjay.
you can read, yeah.
So we do use what are the molecular software they use? Okay, yeah, we do use lots of molecular modeling technologies. Some are internally developed, and some are like basic, what all the academics use. I think Gaussian and docking, covalent, not covalent docking, like usual softwares, covalent docking, usual docking softwares, computational softwares that you use. I believe Schrodinger, that's maybe you are asking. Schrodinger, yes, we do use Schrodinger, lots of Schrodinger platforms for these initial file mining approaches. Won't it be very expensive? Oh, the question, I believe the question is only using the softwares to develop a drug. I know.
No. This is not that. This is Professor Kumar is asking about the synthesis.
Okay,
the large
sorry.
scale synthesis.
Oh yes, yeah. It is expensive. Yeah, you need to have lots of quality controls. it is expensive. And that's why actually the drug has a pricing, to be honest. So we need to factor in all these cost associated with the drug development to the pricing of a drug. So that's why they are that why they are as they. Sorry, I cannot disclose more.
Any more questions? If there are no more questions, it's 4:36. May I invite Dr. Melanie Thomas to propose vote of thanks?
Thank you, sir. Can you hear me?
Yes.
Go ahead.
Okay. So on behalf of the organizing committee of Inspire, I would like to thank Dr. Utpala Seneviratne for this informative and timely discussion you had with all of us today. And we feel very proud to see a product of Peradeniya reaching to such an influential place in the world. And I believe your mere presence inspired all our students to reach their scientific goals. And also your talk made us realize that for a scientist, the only limitation is their imagination. So we are very grateful to you for taking the time out of your busy schedule to join with us today. And we hope you will join with us in our future events too. Let me also thank our dear sir, Professor Andra Wickramasinghe, for inviting Dr. Utpala Seneviratne for today's session and for hosting him today. Thank you very much, sir, for your support and guidance. Last but not least, I would like to thank all of you who joined us today for your active participation. And we hope you found this session very fruitful. Before winding up today's session, I would like to invite all of you for our next week's webinar, another Inspire webinar with our dear sir, Professor O.A. Ideperuma, on 7th of July at 5 p.m. Have a wonderful evening and thank you. I would also like to invite Dr. Utpala Seneviratne to join Professor Andra Wickramasinghe and the students with the Meet the Speaker event using the other link. Thank you.
Use the other link, Utpala, right? We'll spend about half an hour also talking to students as we discuss. We'll close the session now. Thank you.